Welcome to our seminar series, the EEB series. Before I get started to introduce our speaker, I just want to remind you that today at 5:30, from 5:30 to 6:30, we're going to have a mixer for our invited speaker today in the Botanical Garden. It's open to everyone. Hopefully it's going to be nice. We can be outside. We're going to be at the Lacrez Pavilion from 5:30 to 6:30. So for me, it's a real honor and pleasure to introduce our speaker today, Santiago Ramirez, who is visiting us from our sister campus of UCLA. Santiago is an associate professor. He just got tenure 2 weeks ago.
Starting June.
Where he's in the Department of Ecology and Evolutionary Biology in the Population Biology program. Santiago, as you will see in his talk, he's broadly trained evolutionary biologist with really broad interest. But the exciting part about his research, I think that is because he integrates multiple approaches and multiple tools and multiple areas in biology, from phylogenetics to pulp genetics to neurobiology to ecology and also tropical biology. So Santiago's focus mainly is on the evolution and diversification of insects, particularly bees, orchid bees that we're going to be talking about today, but also because he has been doing research in these orchid bees. He's also doing some research now in orchids, also in the evolution of plants in tropics. So Santiago got his bachelor's degree in Colombia at the University of Los Andes, where we actually met many years ago. Then he came to the States, where he got his PhD in Harvard University in the Museum of Comparative Ecology with Pierce. And after being in Harvard, he moved to the West Coast to for a postdoc with Neil Tsutsui Berkeley. And after that, he moved to Davis, where he has been for the last 5 years or so. So with that, please welcome Santiago.
Okay, thank you. Thank you, Felipe. And thank you all for the opportunity to come here and tell you about my work. As Felipe mentioned, a lot of the work that we've been doing, or I've been doing even before I started my lab, has been focusing on understanding the patterns of coevolution between euglossine bees and the orchids that they pollinate. And we've been generating quite a bit of data on both sides of this interaction and the coevolution of these two lineages. But today I will be focusing only on the bee side of things, and it's kind of an exciting time in the lab since we've been getting some newer data that I'm hoping to share with you. And really a lot of the motivations, most of the motivation for our research really comes from natural history, and I consider myself very lucky that we can do that for a living, that we can look at natural history and use the behavior and the species interactions to generate interesting hypotheses and go out and study lineages. So with that in mind, I'm going to focus a little bit on the natural history of orchid bees, because it's really fascinating, and how this natural history has led to a lot of the work that we produce. So orchid bees are for those of you who are not familiar with tropical insects, orchid bees are perhaps the most important pollinator group, insect pollinator group in the neotropics. They're really, really abundant everywhere you go in lowland elevation, wet forest in the tropics. They are very important pollinators of many different plant species. Orchids are just one of the many plant families that they pollinate. The male bees and the female bees forage on nectar from a wide variety of plant species. And in fact, we think, along with stingless bees, they really are the main pollinators of tropical plants when they forage for nectar. Females of orchid bees are also very typical bee, in the sense that they require pollen for provisioning offspring. So that's where the protein comes from, for provisioning to the offspring. And they collect pollen from also a wide variety of plant species and plant families: Fabaceae, Melastomataceae, Solanaceae, and many others. So nothing unique about orchid bees in that sense. But one of the really fascinating aspects of the natural history of orchid bees that has really gave rise to many projects in our lab is this very unique behavior called the male fragrance collecting behavior, or male perfume collecting behavior. And hence they received the name orchid bees, because the orchid bees, the males visit orchid flowers, but also other sources in search for these perfume compounds that they're used in mating behavior. And so, and that's a lot of the work that we've been doing and what I'll be talking about today. So male perfume collecting behavior is a really ubiquitous trait across every species of orchid bees. There are about 230 different species of orchid bees. Every single species of bee exhibits this behavior. This is a video that shows that behavior. So male bees of this Euglossa species identify a flower that is producing a lot of scent, and the male bees land on the flower and they use these front tarsal brushes to collect the scent compounds, and they transfer them and place them in these hind leg pouches, or leg pouches, where they store those compounds. They not only collect them and accumulate them, but they store them for extended periods of time. And they really have very elaborate morphological and physiological and behavioral modifications that allow them to collect and accumulate these compounds. In this case, they're collecting compounds from an orchid flower. So then later on, throughout their lives, after they collect all these perfume compounds, male bees would accumulate them, and then they would soon after that start doing what we call the male courtship display behavior, which is something like this. So a male bee would identify a suitable site in somewhere in the forest, and then start displaying for females, we think, but perhaps other males as well. And in the process of doing this, this is a slow motion or high speed video of that same behavior. The male bees are taking flight. So what they're doing is they're taking little samples of the perfume that they have accumulated in the hind legs. They're transferring that into the middle leg and pressing that against the base of the wing. In other words, what they're doing is they're ventilating or exposing small puffs of the perfume that the male bees have accumulated over time, right? So it's a really unique behavior. Every single species that we look at has this display behavior. Interestingly, in a recent paper that we did, we found out that different species would choose different diameters of trees or different heights above the ground or different times of the day to display. And there seems to be something unique about where to display that seems to attract males of one species to one particular place to display. Over time, when we go to our field sites, we go always to the same trees, and sometimes we find multiple years the same species of bee displaying in the same tree. So there's something unique about where to display that is unique to a species, and we don't know what that is. It could be a combination of sunlight, exposure, and so on. These are some of the morphological traits that the bees are using for that. So this is the hind leg of a male bee with the cuticle removed here. As you can see, basically it's like a sponge. And there's two tiny little openings, well, depending on the species, sometimes one, sometimes two, but that opening is where the perfume samples are deposited, and in these sort of sponge-like structure, the compounds are going to be retained for long periods of time. The male bees use these mid-tibia, so the middle leg has these little hair patches. That's where the male bee places the little samples of perfume when he's going to display, and this is the same mid-tibia, and as he's flying, he would press the mid-tibia against the base of the wing. At the base of the wing, there's this thing called the jewel comb. It's like a little comb that will basically fan out or expose the perfume sample. So the male bee in that video is taking samples from the hind leg, putting it in the middle leg, and pressing against the wing, and then displaying these sort of puff of compounds. So where the male bees get all these chemicals from, and what are these chemicals that they're getting, that's a big part of the research that we've been doing. They mostly fall within three major categories of compounds: the monoterpenes, sesquiterpenes, and aromatic compounds. The common feature of all these chemicals that they're collecting is that they are very low volatility, low molecular weight, high volatility compounds. So typically small molecules, some of them are very common, present in many different plants. If you crush eucalyptus leaves, that pleasant smell that you get, that's 1,8-cineol. Some are also pleasant like vanilla, some are not pleasant at all. In fact, some of these sources are flowers, as I mentioned before, but also decaying vegetation or fungi or even feces or some less pleasant compounds to our noses. But they seem to be able to get these chemical compounds from a wide variety of sources. Orchids, we've done a fair amount of work. Orchids have evolved specialized adaptations to produce these chemicals in exchange for pollination services. So it's a rewarding pollination system. We've been doing a lot of work on that. But I won't be talking much about the orchid side of things today. So mostly what I want to talk about is some of the work that we've been doing to try to understand what are the patterns, the evolutionary patterns that we can infer from looking at variation in the chemicals that they collect and how these vary across the phylogeny of the bees, and potentially get to understanding exactly how this chemical information is being used in mating behavior and how that evolved. So really a starting point for me in this work was looking at the phylogenetic relationships of orchid bees. So during my PhD, I did a phylogeny of the entire group of orchid bees, which coincides very well with what we knew about the general relationships. So there's 5 genera. All of the 5 genera are monophyletic. They form these clear monophyletic clades. And one of the things that I wanted to do once I have this phylogeny in hand is I also learn a little bit about the timing of origin of these bees and learn when these lineages diversified. And for that we could use molecular clock analysis. I don't want to spend too much time talking about this, but I just wanted to mention that we have an idea how old this group is based on fossil calibration. So we could look at the fossil record and look at morphological traits that define specific extant lineages, like this Euglossa that has a very elongated face and a long proboscis. We can unambiguously use this calibration point for the genus Euglossa. We also use honeybees and stingless bees as calibration points. And with that, we got an idea of more or less when the entire group diversified, how old the group is. And we get an estimate between 28 to 38 million years ago, between the Eocene and Oligocene time. So relatively young group, relatively speaking. It's a relatively fast radiation given the large number of species that we have. So now, once we have this phylogeny in hand and an idea when more or less these lineages diversified, then I switched my attention by the time I was finishing my PhD and starting my postdoc at UC Berkeley, starting to learn more about the chemistry of chemical analysis. So I started doing a series of collaborations with this fellow, Thomas Eltz. He's at the University of Bochum in Germany. We started doing a lot of chemical work and asking very simple questions. Okay, all bees, all males of all species of orchid bees collect these perfumes. How different are they? We knew nothing about how variable they were, how species specific they were, and so on. So we asked a very simple question to start with, and the question was, well, are these perfumes distinct or not among different species of bees? So to do that, we use this technique now routine in the lab called gas chromatography mass spectrometry, where we can take a single leg from a male bee and extract all the perfume compounds from that leg and run it through a GCMS, where and the GCMS what is going to do is basically separate all the individual components present in the mixture, and each peak here corresponds to an individual molecule present in the mixture. The relative area under the peak gives us a rough idea of the relative abundance, and in many cases we can successfully give names to these peaks, right? Cineol, 1,8-cineol, very common plant-derived compound, no problem, it's easy to identify. But in many other cases, it turns out these bees are collecting all kinds of weird things that we don't know where they're coming from or where they are, or perhaps nobody ever look at them. So many times we end up with the mass spec, right? Like, so we know that there's a peak, it's a unique compound, and when we fragment that peak into the individual ions, that's what the mass spec looks like. But we don't know what structure it is, but we can cross-reference our own library, and we've been building our own library, so we can say, even though we don't know the name, we know that that peak is present in this B and absent in that B and present in that B, right? So this is kind of the kind of data that went into this analysis. So to ask this question, are these perfume compounds distinct and between different species or not? We went to Barro Colorado Island in Panama, where we sample 15 different species, all in the genus Euglossa, so they are all closely related to each other. And we sample multiple individuals of the same species. We sample them all at the same time in the same forest in an island. So we know if we find differences in the chemistry, we know that those differences are due to actual preference differences, not just availability differences, right? These compounds are being collected. So, and we generate this very, very large data matrix of many different species, well, 15 species, multiple individuals, hundreds of compounds, and ask whether individuals of the same species cluster together or not. Right? We collapse this in a multi-dimensional plot in 2 dimensions, and the answer is very clear, yes. So all individuals, so most individuals within a species tend to cluster together. So if 2 data points are close to each other, that means they're chemically very similar. They're far apart from each other. They're very different. And if you plot this in a 3 dimension and you rotate that, you would really see very clear clusters, very little overlap among species. There's a fair amount of variation in some of the species, but in some others, there's very little variation. So the answer to that question is yes, their perfume chemistry is very species specific. They're not collecting all the same things. Each species is collecting its own combination of compounds. So then the next question that we wanted to ask is, okay, how when we place these distinctive chemical signatures or signals that the male bees are collecting, and when we place that in a phylogenetic framework, what can we infer about the rate of evolution of those traits, right? And so we can analyze this in many different ways, but one way that we came up with was to analyze it using this chemical disparity through time, which is basically you take a phylogeny of 15 different species, those 15 species, the phylogeny is based on DNA, and then we ask the question, okay, what is the amount of, how much disparity do we observe when we step here at the root of the tree? And as we move from the root towards the tips of the tree, how is that disparity going to decrease over time? So the analogy that I always like to use is like, let's say height in the entire room, right? So if we take the whole group, the entire group, that's where we observe the greatest amount of disparity in height. But if we take smaller and smaller groups of individuals, randomly grouped, we would see smaller and smaller amounts of disparity. So likewise, we progress from the root of the tree towards the tips of the tree, and the expectation is that the amount of chemical disparity contained in these smaller and smaller groups is going to decrease over time as we move from the root towards the tips of the tree. And we can use that to generate a null model, right? And depending on what null model of trait evolution you use, you get different answers. But in this case, we use Brownian motion model, which is perhaps not the best model, but is the best that we have, at least for this kind of data. So the idea is that for this given phylogeny and that given chemical data set, we would expect that the chemical disparity should decrease over time in this fashion. And we can compare that null model against our actual observed data in that phylogeny with that particular community. And if the chemical trait data falls above the null model, that suggests that chemical traits are evolving at a much faster rate than expected under a null model. Otherwise, if the empirical data falls down here, that would suggest that those traits are evolving at a much shorter rate or slower rate than expected under the null model. And what we find is that throughout the phylogeny of those 15 species, the chemical traits seem to be evolving at a much, much faster rate than expected under a null Brownian motion model, suggesting that these chemical traits are really, really evolving very fast among sister species, right? When you're looking at very close related species, the chemical traits seem to be divergent very quickly away from each other. Now, that was a study that we conducted in a single community, 15 species, and since then we've expanded this into a much larger analysis. And former postdoc in my lab, Marjorie Weber, who's now at Michigan State University, she managed to analyze this really large data set of 65 different species, all in the genus Iglossa. But interestingly, the individuals that she analyzed came from many different communities. So she was not only analyzing from a single community, but from multiple communities. And there are many interesting details about this study, but the main result really is that we find exactly the same pattern. Across the phylogeny of those 65 species, we would expect chemical traits to evolve in this fashion, but throughout the phylogeny of those 65 species, we observe a much faster rate of evolution, especially concentrated towards the recent. So sister species seem to be evolving very quickly away from each other in terms of chemical space. A different way of visualizing this would be in this beautiful figure or video that Marjorie produced. Here in the horizontal plane, you have chemical disparity for different species. So each data point corresponds to a species. If the 2 points are close to each other, they're similar. Far away from each other, they're very different. And the lines here represent the phylogenetic relationships of all those 15 species. And as you can see in this rotating video, you have things that are very close to each other, phylogenetically speaking, but chemically very far from each other. So very quickly after speciation, basically chemistry goes away from each other. An really fascinating result that came from this analysis that Marjorie conducted was she asked, because things seem to be evolving very quickly early on after speciation. So she asked, how is this rate of evolution maintained, or is it decreasing as the species diverge from each other? And so we only had a few of those 65 species, we only have a few cases where we have sister species for which we had very good quality data. And we could ask questions about the rate of evolution, so how fast chemical differentiation is evolving over time, so the rate of chemical evolution as a function of how divergent those species are. So if the species just form, these two species are very young, the rate of evolution seems to be pretty high. And as these two species starts to diverge away from each other, the rate of evolution seems to decrease over time. It's a really fascinating pattern that seems to point in the direction of something is really driving chemical traits away from each other. And the immediate hypothesis that we came up with here is, we've been thinking a lot about this for a while, is that perhaps it's something like reinforcement, right? Where if two species just diverge and if they have the potential of hybridizing, and those hybrids have a lower fitness, then natural selection should favor the evolution of a signal that is far away from each other. So reinforcement has been demonstrated in many systems, especially in communication systems, but this seems to kind of hold, it doesn't demonstrate it, but it seems to go along with those lines. So maybe natural selection is really pushing these chemical traits to evolve away from each other, and as the two species diverge away from each other, the selective pressure is lower. Interestingly, a corollary of that would be, well, you would expect that kind of rate of evolution to be higher in sympatry than in allopatry, and sure enough, that's what we find. It's a correlation, but it goes in the right direction. So sympatric sister species seem to have an even higher rate of evolution, which is, again, not a demonstration of reinforcement, but at least suggests that could be at play. So one thing that I didn't mention that is just really fascinating about the natural history, the details of orchid bees, is that when these bees are collecting chemical compounds from the environment, they're dealing with very volatile molecules that are really hard to keep in place, right? These are molecules that would evaporate in seconds if you put it in a piece of paper. So the bees have these very large tibial pockets, and they have these sort of sponge-like structures but they also have these very enlarged labial glands in their head. So all males of all species have these. Basically, if you open the head capsule, you see the brain, but in between the brain and the cuticle, you see these very thick layer of glands full of lipids. And what the males do is when they land on a surface and they collect these chemicals, they basically spit all these lipids into the flower, into the surface, mix that up with the perfumes, the compounds, and they transfer that mixture, put them in the hind legs, and that basically serves as a solvent, right? Like those lipids are maintaining those chemicals in the leg. So whenever we extract a hind leg from a male bee, we find the perfume compounds, and also we find all these lipid molecules that we know come from the bee. The bee produced them. And so we can separate them most of the time. We can separate them very well. So we conducted this same analysis that I showed you before with perfume compounds. This is the same thing, rotating figure, but looking from above. Species connected by their phylogenetic relationship. So these two species are sister to each other, yet chemically they're very, very different from each other. That's based on the perfumes. So if we conduct the same analysis with those lipid compounds, we find a very, very different answer. Like in most cases, the composition of those lipids doesn't seem to change, seems to be kind of constant. There are a few cases where sister species do have very distinct lipids, but for the most part, the perfume compound, the lipid compounds don't seem to be evolving at a faster rate than we would expect, than we observe in the perfume compound. So there's a lot more phylogenetic signal in the lipids than there is in the perfume compounds. So you could not predict that these two things are sister species based on chemistry. That is interesting because it is telling us that even though the bees are adding stuff to those chemical compounds, perhaps they're only serving as solvents and not necessarily as actual mating signals or mate recognition signals or sexually selected signals. We think these are just solvents. In some cases, they might serve a function. We don't know. But this is a really sort of interesting comparison that Marjorie did when she compared the acquired compounds versus the bee-produced compounds. So just to summarize this part, these perfume phenotypes appear to be highly, highly divergent among bee lineages. And even though it's a correlation, these patterns of perfume divergence point into the direction of perhaps reinforcement. And to really demonstrate the reinforcement, we would really have to do very careful experiments. Some of them might be hard to do, but really what this is telling us, what this is pointing to, is that perfume chemistry appears to be very important in mate recognition or sexual selection, or both. We don't know which one of those, or it could be really both. But we really know that they seem to be very important in also in the early stages of. It seems to be evolving very quickly in the early stages of divergence, suggesting that they could be quite important in perhaps in the formation of pre-zygotic reproductive barriers among species. So really the next project, and this is what we've been very busy with, is trying to understand what really happens early in the early stages of speciation of different species. So for this, we ended up focusing on this one species. This is kind of an interesting side story. Euglossa viridissima was a single species broadly distributed throughout Central America. And somewhere hidden in the taxonomic literature, someone described, well, yeah, this species appears to be polymorphic in terms of number of teeth in the mandible. Some male bees have 2 teeth, some male bees have 3 teeth in the mandible. But at that time, the taxonomic treatment simply say this is a polymorphism, nothing else is different between these bees, so no need to separate them as different species. And it was exactly one line in that taxonomic paper written in the 1970s that really inspired a whole set of projects after in my lab and my collaborators' lab. So we call these the 2D, the two-dentition or the three-dentition bees. When we started looking at the chemistry, just in the same way we look at before, we found very quickly that there are two very distinct chemical phenotypes. The male bees smell like this, or they smell like that, but really there isn't nothing really in between. When we look at what happens with the number of teeth, it almost makes all perfect sense, but it doesn't quite make perfect sense. So if the male bees have 2 teeth, which is the common phenotype, they would fall in this cloud of individuals. If they have 3 teeth, mostly they fall into this cloud of individuals. There are a few individuals here in red that also have 3 teeth that fall in this kind of sort of the wrong part of the cloud. So we start digging deeper and what was going on. Do we have 1 or 2 species? Do we have a polymorphism or not? Then when we start looking at carefully what goes on here, we found that all these individuals here, they both share some compounds, but the main difference between them is that these individuals collect HNDB, is a novel molecule that we now have the structure for, and these individuals here don't collect that molecule, but mostly have another molecule called lactone 97. We don't have the structure for that molecule yet. And when we look at the number of teeth, well, it mostly it falls in this fashion. But there's an interesting thing. Those individuals that fall into this cloud that have 3 teeth, the distance between the apical and the middle tooth seems to be much shorter than the individuals here. So at the time, this is 2011, we also did microsatellite analysis. We look at every single morphological trait. Male genitalia in insects evolves very fast, so we look at it exactly the same, no difference at all. The only difference that we observed were on the teeth, and the chemistry was very clear. So at the time we decided, well, this is we're going to name this a new species, Euglossa dilemma, Euglossa viridissima. The genetic data that we look at made sense at the time. So I guess that taxonomic report in the beginning was correct in saying that yes, there is a polymorphism in the sense that this species is polymorphic, but this one is actually not polymorphic. But the chemistry seems to be always holding true. So my student, Philip Brandt at UC Davis, has taken on this project, and done a really detailed work on trying to understand what goes on in these 2 species. So using museum records and his own field collection, now we have plotted the distribution of the 2, what we call the 2 different species. Euglossa dilemma, a little bit from the south, it goes from Costa Rica all the way to Veracruz in Mexico, and Euglossa viridissima goes from northern Mexico down to Guatemala, northern part of Guatemala. So it's a really neat sort of distribution. We have allopatric populations here and sympatric populations here in the middle. And using these populations, we've been doing a very detailed work on population genetics, but also on chemical analysis of the perfumes. And in addition to that, some whole genome resequencing. So we use genotype by sequencing. So basically, you take anonymous markers distributed throughout the genome. This is a chromosome. And you sample those same anonymous loci across multiple individuals to build a population genetic analysis. We conduct the perfume analysis the same way I indicated before. And for a select number of individuals, we conducted transcriptive analysis or whole genome analysis to ask questions about the genomic basis of this perfume differentiation. So I'll get to this later. But for the first part, I want to show this data, the GPS data. So this is a lot of work, but one figure that summarizes everything. Euglossa viridissima, the one that is up here in the north, the one that does not collect HNDB, the one that collects lactone 97. That species seems to cluster very tightly in this corner of the plot. And dilemma, on the other hand, Euglossa dilemma, the new species that we described, seems to have a lot more genetic variation than viridissima, to the point that it almost makes you think of maybe there are even 3 species, or maybe there is only 1 species. And so we kind of go back and forth. We're still, with the current data, we're pretty convinced that these are 2 distinct species. And we call them viridissima and dilemma. But the interesting thing is that there is very little genetic differentiation between these two species. If you look at PC1 and PC2, it's really only explaining very small amount of the variation between these two. So that kind of reinforces what we suspected. These are two recently diverged species, very recently diverged. And Euglossa dilemma has even greater amount of genetic variation than there is between dilemma and viridissima, which is quite interesting. So when we look now at low levels of genetic diversification, what I said already, when we look now at the perfume chemistry, we get a pretty similar picture as I indicated before. We have basically two clouds of compounds, or two clouds of individuals, I'm sorry, based on whether they have HNDB or lacto-97, and pretty much correlates with the genetic data that I just showed. These overlaying circles are quite important actually, because the diameter of the circle here correlates with the total amount of perfume that a male bee has. So because these are acquired traits, that the male bees are going out in nature collecting these perfumes. If a male bee just emerged and he starts to collect perfumes in the first day or second day, he would only have a very few sort of defining chemical compounds. So here in the middle, as you can see, most of the individuals that we sample tend to have very few amounts of perfume. And as they get older and they have more opportunity to collect their species-specific perfume, they seem to be pulled into sort of the species average, I guess, if you will, of the perfume chemistry. So when we look at only older males, we see really very distinct differentiation. The younger, more naive males seem to kind of fall here in the middle. But doesn't matter how we look at this data, the presence of HNDB in dilemma and the presence of lacto-97 in viridissima seems to be pretty much the best defining character, other than the mandible. The mandible trait is really hard to look in the field, or even under a microscope. So just to summarize this part, this seems to suggest that we found a very recent speciation event where there are very low levels of genome-wide divergence between the sister species, yet the chemical phenotypes are very species-specific, and that specificity really is independent of where you sample them across the geography or how much population structure we observe. So now where we're taking this project to is asking the questions of how that divergence happens, and how you go from having an ancestral species into two daughter species that are collecting slightly different set of perfume compounds. So we've been attacking this from multiple angles, but my colleague Thomas, back in 2008, conducted a very simple experiment that kind of really inspired subsequent studies. And the experiment is the following. You can take the antenna of a male bee. In this case, there were a few females, but mostly male bees. For reference, these little hairs are the sensilla. Each of those sensilla contain the olfactory receptor neurons that insects use to smell. And you can take a whole antenna from a bee and probe it with an electrode and present that antenna against a variety of compounds. And if you see a strong action potential from the entire summation of all the sensilla being activated at once, then you would know that bee has a very highly tuned sense of smell for that particular compound. If it doesn't respond, that means it doesn't have the receptors for that. So when we present, or when Thomas presented Euglossa dilemma and Euglossa viridissima to a hydrocarbon that is sort of a standard that is not specific to any bees, the two species, the males of the two species, seem to have a very similar response. When we presented Euglossa dilemma to HNDB, they responded very strongly because that's the compound that they collect, whereas Euglossa viridissima does not respond to HNDB. We haven't done this with lacto-97, but you would expect the opposite. So that really led us to think, okay, there's something in the sensory system of male bees, in the antenna level, not just the central brain, that controls the perfume specificity, at least part of it. At least there seems to be a sensory adaptation to be highly tuned to collect or not collect certain compounds. This is just a quick summary of, okay, we have all those sensory hairs or sensilla in the surface of the antenna. Within each sensory hair, if you take a slice, you would see these olfactory neurons. Each of those olfactory neurons would express olfactory receptor genes. That those are proteins that bind, or to which ligands or odorants bind to and activate that neuron, and those signals are sent into the antennal lobes and to higher level brain centers. So because we know from this experiment that there's something in the olfactory receptor neurons that is different between the species, we really wanted to know how different the olfactory receptor genes are between the two. So we took a transcriptomic approach in which we tried to sequence and assemble all the olfactory receptor genes expressed in males of the two species. There's also a smaller family called the ionotropic receptors, also involved in the detection of acids and some other molecules, and gustatory receptors. They can taste through the antenna. They can taste sugars and so on through the antenna as well. So when we conducted these transcriptive analysis, we assembled these 3 gene families, but mostly we were interested in the olfactory receptor genes, these 7 transmembrane gene families. So when Philip did that, we basically created a large gene family tree for the olfactory receptor genes for Euglossa viridissima, Euglossa dilemma. I'm happy to tell you about all the details that went on into these, but basically what we wanted to ask is, we have all the set of olfactory receptor genes in dilemma and viridissima. We can annotate them and reference them with the honeybee, which has a very high quality genome, and then ask questions about rate of evolution in those orthologous genes that are shared between those. And so in some cases we do find, very, very clear cases of orthology where we have dilemma and viridissima sister to each other and those sister to honeybee. We focus on those and use those to ask questions about rates of evolution, and rates of amino acid substitutions. And so we ask the simple questions: are any of these orthologous gene copies exhibiting a higher rate of non-synonymous substitutions over synonymous substitutions? That's the standard omega value there. And we actually did identify 3 candidates, or 3 interesting orthologous gene copies that seem to be evolving at a much faster rate than expected under a neutral model in these 2 species. They seem to be accumulating more non-synonymous substitutions. This is the same data, basically. So we plotted the number of non-synonymous and synonymous substitutions, and we identify these 3 gene copies that seem to exhibit high rates of amino acid replacements. In particular, we were very excited about OR41. It has 17 or 18 different non-synonymous substitutions between the 2 sister species of orchid bees. So back in 2015, we kind of thought, okay, maybe OR41 has something unique between these. So let's look into more detail. So to step back a little, so when we have these population genetic analysis of the different individuals, we took a few small subsets of individuals for which we conducted whole genome sequencing to ask questions of divergence across the entire genome, and in particular in these olfactory receptor genes. So we took individuals of dilemma from here, individuals of dilemma from here. We didn't take from the St. population yet. We haven't done that yet, but we will soon. So we took from the allopatric populations, and we conducted this genome-wide analysis where we have an assembled genome of dilemma, and we ask how divergent is the genome, or in windows of 50 kilobases, how much divergence that we observe across the genome. It's a relatively good assembly, but in very many contigs. So each of these bars corresponds to a scaffold, so a stretch of the genome, not a chromosome, but rather a long stretch for which we managed to assemble. And we ask how much differentiation there is across the genome using this common metric, FST. And not surprisingly, this has been found in many other recently diverged lineages. You find some regions, some islands of high divergence across the genome. that are interesting, that may harbor some interesting genes, so we'll be looking closely at those. FSD is a relative measure of divergence. There's another metric called DXY that is rather an absolute metric of divergence. And interestingly, we find cases where there's high divergence in FSD but low divergence in the DXY. We're still making sense of this data, but looking through the literature, it seems to be that this is potentially a case of purifying selection. So background selection very strongly happening in these two species. And as this background selection eliminates allelic diversity, and after the species diverge from each other, you may end up with highly differentiated regions of the genome. But there's nothing unique about that. It just simply happened to be that there was a reduction in allelic diversity and those regions became differentiated. And it doesn't mean that there was a strong directional selection or anything like that. We're still trying to make sense of this data. It just came out recently. But when we look at the two metrics, both FST and DXY, and we plot this in this plot, we find that there's a lot of regions that have very high FST but low DXY, but there are a few interesting candidate regions here that are high for both FST and DXY. When we look at those, interestingly, two of those fall into this scaffold here, which is a scaffold number 61. That's the scaffold 61, where this is the FST plotted. And scaffold 61 is precisely the scaffold that contains OR41. OR41 is here. Olfactory receptor genes are arranged in a very unique way in the genome. They're arranged in clusters, so tandem arrays, recent duplications, or ancestral duplications. All these red genes here are olfactory receptor genes, and OR41 happens to be right in the middle, right there. So again, OR41 emerges as an interesting, highly divergent candidate between these two species. When we look at the amount of allelic diversity within each of the species, pi, within the Viridissima and Dilemma, we find that Dilemma has much lower allelic diversity than Viridissima, suggesting, and this is again correlational, suggesting that if there was strong selection in one of these olfactory receptor genes, it was in the lineage leading to Dilemma, not necessarily Viridissima. But this is again correlational, right? And we've done a little bit of targeted sequencing of these particular olfactory receptor genes to gain more insight of what's going on between them. And OR41 keeps surprising us. So when I mentioned we have allopatric and sympatric populations, and in Viridissima for OR41 we only have one allele. For Dilemma it turns out that there's two alleles. But interestingly, and when we do the phylogeny, it actually suggests that a lot of the changes occur along the branch of Dilemma, not necessarily Viridissima. And when we do a detailed analysis of all the alleles present in Dilemma, we find that actually the two alleles pretty much perfectly coincide with whether they come from a allopatric or a sympatric population. So suggesting that in the allopatric population we only find this one allele, and these two AB alleles we find only in the sympatric population, which doesn't demonstrate anything, but again makes us think about perhaps something goes on in the area of sympatry. Perhaps if there is some kind of reinforcement, strong selection to smell different and cue on mates that smell different, that happens more strongly perhaps in the area of sympatry. But again, it's correlational. So we don't really have a strong demonstration for reinforcement here, but it's a really interesting pattern, at least at the allelic level here. Other interesting results from this is when we look at the gene trees that we can recover, or the gene phylogenies that we can recover with OR41 versus other different genes that we sequence, is that many of the other olfactory receptor genes do not recover the species phylogeny. So in this case, there's very little divergence between them. In this case, dilemma and viridissima cluster together, or they cluster together like here. But in the case of OR41, it does perfectly recover the species tree, or what we consider the species tree at least. So again, this kind of suggests that perhaps this OR41 is playing a very critical role in reproductive isolation, and we don't know the ligand that activates this, of course, and that's one of the directions we want to go into, is to try to identify the particular other molecule that is responsible for this. The obvious candidate for us is HNDB. HNDB is the compound that the glossal dilemma collects, but until we have actual proof of that, it's just conjecture. This is just a quick overview, just a model of the olfactory receptor genes, the different copies that we found, and where we found the different non-synonymous substitutions in the amino acid sequence. As I mentioned, OR41 has 17 or 18 different substitutions, and they happen really in the transmembrane domains, but also in the intracellular and extracellular domains, which makes sense if there's any change in the configuration of the protein. Any of these amino acid substitutions could lead to such changes, and in fact it has been shown in moths and in flies that non-synonymous substitutions anywhere really along the protein could actually render a change in the sensory perception of those receptors. So just to summarize this part, these bee perfume phenotypes are very distinct among recently diverged lineages, and it appears that this olfactory sensory system is perhaps implicated in pre-zygotic reproductive isolation. Again, not demonstration, but it all points into that direction. And if that's the case, it's quite possible that those early changes in sensory perception could favor reproductive isolation and the origin of new species. So with the few minutes that I have, I just want to mention what we're doing now and how we're trying to tackle the problem of knowing the functionality of OR41. This is we're spending a lot of time and resources in trying to get to this, but basically what we're doing is taking Drosophila flies that you can very easily order knockout lines that have very specific set of receptor genes that don't have fly olfactory receptor genes, so they're removed. So they're called the empty neuron because those neurons are not responding to anything. And then you can insert your olfactory receptor of choice of any insect into those neurons. And so you can, once in the fly, you can conduct a very relatively easy neurophysiological recordings. We punch them with an electrode, expressing your B receptor of choice, and present them again with a set of order, and you can measure the actual action potentials of those neurons, and ask questions about the tuning of how broadly tuned or narrowly tuned these receptors are. So this is how it looks great. It records beautiful. We've been able to do wonderful recordings. For whatever reason, the B receptor genes are not functioning properly as we would expect once in the fly. We've been able to clone other fly genes and even Lepidoptera genes, so the system works. Something unique about the B olfactory receptor genes, and we don't know what it is. So at the end of the day, what we want to do is create sort of a tuning curve, if you will, for these different alleles. So we know what the different substitutions are, the different alleles of B, ORs, and then we can ask questions: Are some of these genes narrowly tuned to one compound or broadly tuned to different compounds? And what happens every time that a new substitution is introduced? Is there a change on the tuning or a new function for that OR being evolved? We have a number of behavioral experiments too going on with Euglossa dilemma. Euglossa dilemma is in the US now. It was introduced 20 years ago in Florida, accidentally, and It's actually very abundant and pollinated a lot of invasive plant species, but so far they haven't tried to eradicate it. And so we're taking advantage of that. And then at the University of Florida, we have this large enclosure where we can mark individuals, follow them, and we can manipulate their perfumes and do more functional behavioral experiments. With that, I wanted to thank, of course, everybody in the lab, the UC Davis, and funding sources that really help with a lot of this. And thank you for listening. Yes, question.
Fascinating talk.
Thank you.
So I was thinking, the reinforcement, I think you make a good case for it, but I was also thinking in terms of character displacement. And, looking maybe beyond sister taxa, but, since these perfume, spatially can be so different, do you have instances where there's evidence for character displacement where you have two species coming together?
In that case, character displacement would be something ecological, not necessarily involved.
Well, with the perfume in the perfume space. In other words, do you find that they're more divergent? The perfumes are more divergent in sympatry than allopatry?
Yes, yeah, we do find that in some cases. We do find that across the whole phylogeny tree analysis that we did. When we look at the Dilemma and Viridissima, those two sister species, we were asking that same question: Are the perfumes any different in sympatry than in allopatry? And the answer is no, they're the same. Which is so. I guess the reinforcement argument works only halfway, right? Like it works halfway in the sense that perhaps there is reinforcement at the molecular level to change the receptors to make it more efficient at finding HNDB or whatever compound is, but the trait doesn't change. The trait remains the same. The classical case of in frogs and reinforcement is that, in areas of sympatry, the actual song or the call diverges to prevent hybridization. But in this case, the perfumes remain the same. So, yeah, I don't know if that answered your question.
More or less. I was just thinking if there could be, in cases where you had, two species coming together, it could be, with the same signal, it would be a loss of fitness for those species having the same signal in the same place.
Yeah.
Potentially.
Yeah, and we keep finding, I guess, sort of independent suggestions of that being at play, but to really do it properly, I think you would have to do transplant experiments, and we haven't got there yet. Yeah. Yes.
Well, you can also have greater divergence in mating recognition in sympatry than allopatry. Maybe that is what you're seeing with the different receptor levels. I guess you don't know what phenotypically those effects are or having on female, discrimination between different perfumes.
Right.
Potentially, they could be better because of that difference, better discriminating between the two perfumes in sympatry and allopatry, which would result in greater.
Right. Yeah, so you raise an interesting point, and so I guess I didn't mention is in this case we have both the male trait, so the perfume, and the female preference potentially also both male trait and female preference controlled presumably by the olfactory system, right? So you could envision a situation where a single mutation could affect preference by the females, but also the male traits by the males.
What I suggest is maybe the male trait is there's no greater difference in the male trait in sympatry than allopatry because they're totally distinct.
Right.
In both cases, but it could be that females are better discriminating between the two trait values in sympatry than allopatry.
Yeah, exactly. Yeah, and what we're looking at right now is only the males, right? Like we're assuming we're only measuring the male, but we could be measuring the female, more choosiness of the female. But the fact that they're using the same gene for both male trait and female preference could be the reason why we're interpreted that way. So I can talk to you more about that. Yeah. Question there. Yes.
So I'm really curious about the area of sympatry.
Yeah.
From the point of view of whether they like the species or not. So if you run a mass structure mixture analysis. Do those ones in sympatry show up as hybrids, or do they show up as?
Yeah, I do have that figure. Again, it just came out, so good time to ask that question. Here it is. So, no. Oh, I have it hidden, so let me unhide it. So we do this structure plot. So
this is regular structure or fast structure?
This is regular structure. Yes.
How many markers?
How many markers we use? This is with the GBS data, 15,000 SNPs or so. Yeah. So when we run a k equals 2, Viridissima and Dilemma, for the most part, they're not separated, and it's the first separation that appears is within Dilemma. And St. Patrick, it says St. Patrick here, goes from here to here. So at least some of the St. Patrick individuals may appear as hybrids between these two. Then when we have a k equal 3, then the separation of the two species happens. And again, I guess it shows, it's trying to separate basically the St. Patrick species into a different species. Yeah. Yeah. So maybe
Is equal to favored, you know, in terms of
in this case, k equals 2 is favored. Yes, I remember correctly. Yeah. Yeah.
it doesn't look like from that point of view there's real support necessarily for those two species.
Yeah, not really, and it comes from the fact that Dilemma has greater amount of variation than between the two species, really. And interestingly, I guess I didn't mention this. Like, yeah, I mean, most of the area of symmetry happens in Yucatan Peninsula, so yeah, this is mostly the area. But these guys here in Veracruz actually are.
Is that low elevation Veracruz or high elevation Veracruz?
From sea level to 900 meters, they occur there. And I don't know exactly where. Yeah, so the interesting thing is that these individuals here are more similar to the allopatric population than they are to the individuals here in the allopatric. So now we're thinking there's something weird, unique. It's almost like there's like a major bottleneck of some sort, or some sort of, or a lot of genetic drift going on here, but it doesn't make sense. I mean, why would you have such genetic drift here? Or perhaps the population was constrained here and then colonized those two regions. I mean, it just doesn't make sense. Like we haven't been able to kind of think of a biogeographical scenario that kind of explain why you would have these individuals so closely to that and not to these. I mean, it almost makes you, this whole thing here is mostly Yucatan, it almost makes you think that's a separate species. But there's nothing unique about perfume, nothing about any OR. It could be a cryptic species that is maintained by something else, or it's just simply population structure. I mean, where you draw the line here is really hard. Yeah. There's a question. Okay. Yes?
I was curious if you've done any collected media or done some real experiments to see if the different bee species are favoring different flower types,
Yeah.
or if there's some kind of like pollination, mutualism that is unique to these two. Does it come out in their flowers?
Yeah, no, that's a great question. So I didn't talk at all about the work that we've been doing on the bees, on the plant species that they visit. So as I mentioned, they collect these perfumes from orchids, but also from fungi, all kinds of weird sources. In the past, people think, oh, they were only getting them from orchids, and therefore, if you visit one species of orchid, then your perfume is going to look like that. But it turns out that the orchids are only providing a very small fraction of all the compounds that a bee needs. So, if you rank all the compounds that a bee collects, it's 40 or something like that, and of those, maybe 1 or 2 may have come from an orchid, but the majority of those came from wherever they can find them. They're really good at finding a leaf that is infected with fungi or bacteria, and they find that compound and collect it, and they go elsewhere and do the same thing. So in the past, yeah, we would think, oh, maybe the orchids that they're affiliated with have a huge influence in the perfume, but the more we look into it, seems like they just collect whatever they need, and if it's from an orchid or a fungus, That's all they care. Yeah, they don't care. Yeah.
Right, so just thanks Santiago again.
Yeah.